Hello everyone. Today we'll discuss the another application of recombinant DNA technology, that is production of vaccine by using the recombinant DNA technology. So first of all, what is vaccine? It is any preparation that is intended to produce immunity against a disease by stimulating the production of antibodies. Means the vaccine that includes the what? That's maybe the suspensions of killed or attenuated organisms or microorganisms or product or derivatives of microorganisms. So the most common method of administrative vaccine is by injection, like the what that is a vaccine of the corona, that is a Covaxin, Covishield, etc. But some are given by mouth or nasal spray, like the what the polio dose given by the what that is orally. So this vaccine is any preparation that brings about the immunity against any disease within the body by stimulating the production of antibodies. Then these vaccines are antigenic preparations that on injection to the bloodstream stimulate the immune system to synthesize antibodies that protect the body against subsequent infection. Many infectious diseases have been brought under control in the present century as a result of vaccination programs. The vaccines normally contain the inactivated form of the infectious agent as the antigenic material. Heating or similar treatment is used for preparing the vaccine so that the virus particles become attenuated or they are no longer infective or they become the harmless. The inactivation process must be 100% because in this attenuation, even one live virus particle that can cause the infection. Many virus particles for the production of vaccines are obtained from the tissue culture. However, some viruses do not grow in tissue culture. Example, that is a hepatitis B virus. So this recombinant DNA technology can overcome the problems for the production of the vaccines. The key finding has been that isolated component of virus, that is the what proteins of the virus code, can also serve as antigens to elicit the synthesis of virus-specific antibody within the human body. So the antibodies bound to virus after infection inactivate the virus. Based on this principle, there are two methods of production of recombinant vaccine. First is the cloning in the expression vector, and second is the production of recombinant Vaccinia viruses. How this vaccine is produced by the using this Vaccinia virus we'll discuss. The recombinant Vaccinia virus, that is in 1976, the Jenner discovered the Vaccinia virus, and this Vaccinia virus it is supposed to be harmless to the man and can stimulate immunity against the smallpox. And this discovery ultimately led to the eradication of smallpox in 1980. The use of Vaccinia virus as a carrier agent in recombinant DNA technology has made the vaccine production more easier. The Vaccinia virus has been used because of its large genome, which can accommodate near about 25,000 extra nucleotide, and and it is a non-infectious one or non-harmful. The vaccines prepared by cloning of genes for viral surface antigen that elicits the immune responses that is shown in the figure. The viral pathogens have the what? The surface glycoproteins. What you are doing? Here we are isolating the virus code protein. This is the virus code protein. This is a virus. And we are isolating the virus code protein. And directly this virus, instead of what injecting the virus or attenuated virus, we have isolated the virus code protein, and what we are injecting after isolation of the virus code protein, we are injecting it into the what that is the body of the person. What will happen against this, as this is the what only the what virus code protein, and so that against this virus code protein body will form the antibodies, and such type of the what the antibodies specific to these code proteins will be formed. And next to this, these antibodies, whatever the antibodies which are present within the body, what they will, what these will, the antibodies which are present in the blood, they will attach to the what this antigen or this virus. The antigen antibody reaction will occur and it will inactivate this virus. So this is how use of isolated virus protein as a vaccine. Okay, but this functions as an antigen and elicit the production of antigens in the B lymphocytes of the host. The hepatitis B major surface antigen gene has been ligated into the Vaccinia gene. This is the another method how the what that is the production of vaccine can be done by using the recombinant DNA technology. Here, what you are doing, this is the first method, that is a common method, but by using the recombinant DNA technology, what we are isolating the major surface antigen gene from the hepatitis B virus. This is the hepatitis B code protein gene. This is the gene for the hepatitis virus that we have isolated and inserted into the what that is a vector molecule along with the what that is a vaccinia promoter. This vaccinia, it is ligated in the genome under the control of vaccine promoter, and vaccinia particles containing the recombinant vaccinia genome were injected into the bloodstream. The new vaccinia particles are formed, and also the hepatitis B major surface antigen protein, and this stimulates the immune system to synthesize the antibodies against both vaccinia and the what that is a hepatitis B, because here the what anti vaccinia antibodies are raised on the surface, and these are the what the this is the what anti vaccinia antibody on the surface, and the what that is a anti hepatitis B antibody is also there in this. So this therefore immunity against the what that is a both smallpox and the what that is a hepatitis B virus will result. So this is how the what the same thing, that how you are adding, this is the vaccinia promoter and this is the hepatitis B code protein gene that have incorporated, and next to that the what the recombinant vaccinia particles, these particles injected into the bloodstream, and so that what will happen, this virus is a what the harmless, but the anti-vaccinia antibody will produce, and the anti-hepatitis B antibody will be also produced. So the synthesis of antibodies by the immune system will occur. So here the what how this is another vaccine, that is a vaccine against the herpes vaccine. Here this is what the this DNA is extracted from the what that is a this simple herpes or the herpes simplex virus, and this is a gene, that is a red colored gene, which is isolated from the simple simplex, that is a what herpes simplex virus, and this is a, such a virus which is a not harmful, that is the, and it is this genome is what that is a the what such type of the what the vector molecule, this is the DNA from the vector, and this is a what the desired gene, that is from the what herpes simplex gene. Now the what this is a recombinant DNA molecule. And this recombinant DNA molecule, this is a harmless Vaccinia virus and it having no surface antigen. When you are adding the what this recombinant DNA molecule, what will happen? The this will the what that is a fragment containing the surface gene combines with the what the cleaved vaccinia virus, and you can observe the what that is a these surface antigens which are present on the what that is a this vaccinia virus, and this is a harmless engineered virus with a surface like the herpes simplex is injected into the what human body, and this such type of the novel virus which is a harmless but having the antigen of the what this herpes simplex virus, it will produce the antibodies within the body of the human against the what that is a herpes simplex virus. So simply what you are isolated the what you have isolated the desired gene which is what expressing this surface protein, and you have cloned it into the what that is the DNA for the what to make the what the recombinant DNA molecule. This is the vector for the vector and or vector DNA, and what this is the what the recombinant DNA molecule, and this recombinant this is the what the and you this is the what herpes, this is the entire genome of the what that is the herpes simplex is isolated. Then you have isolated the gene by restriction endonuclease, then isolated the what that is this what the DNA from this what vaccinia virus, and then we have ligated the what this gene of the what that is specifying the surface protein of this herpes simplex virus into the what the DNA of this, and then again incorporated into this, so that what will happen this protein will express itself, and for the there will be the production of the or the formation of the what this such type of the what that is the surface protein on the what the harmless vaccinia virus, and such harmless or novel harmless genetically engineered virus is now injected into the body of the human, and so that our body will respond at it as a foreign particle, and it will produce antigen antibodies against the what that is a herpes simplex virus. This is how the what you can produce the what that is a vaccine by the genetic engineering technology. Likewise, the recombinant vaccinia virus expressing a number of foreign genes have been also constructed. Here, the what that is a this is a what this virus will produce only the what antibodies against the herpes simplex virus, because you have ligated the only gene specifying the herpes simplex surface protein. Likewise, you can add more than two or more than two or three or like that, and that can mean single antibody that or the single antibody will confer the what that is a resistance against the what many diseases. Here what you have taken the what that is a vaccinia genome which is a harmless, this is the genome of the vaccinia virus, and this vaccinia virus, and it having the what this rounded surface coat protein or the surface glycoprotein. Then we have isolated the what the gene, desired gene from the three again different virus. That is hepatitis B virus. This is the surface protein for the hepatitis B. Then this is a what herpes virus, surface glycoprotein, and this is a what influenza virus. These are the surface proteins of these three different virus, and this is the surface protein of the vaccinia virus. We have isolated the genome of the vaccinia genome, we have isolated the gene which is expressing this protein from the what that is a hepatitis B virus. This is a gene. Then we have isolated this gene which is expressing the what herpes virus surface glycoprotein. And again we have isolated the desired gene from the what influenza virus which is expressing the what such type of the glycoprotein. Now what you are that ligating all these three genes with the what? The vaccinia gene. This is the gene for the what? That is the what? Influenza, then herpes and the hepatitis. And then entire after ligation, this vaccinia genome with integrated glycoprotein genes of different viruses are now introduced into the what? This Vaccinia gene. The surface glycoprotein of different virus will appear on the Vaccinia virus coat. That if you observe this T-shaped, that is a what? It is a what surface protein for hepatitis B virus. Then this is for the what? The influenza virus. Then this is for the what? The herpes virus. And this rounded or the small, that is for the Vaccinia virus. So this is a what? The polyvalent vaccine that having the what? The surface glycoproteins of different viruses. This is a harmless one. This is a, as it is having the Vaccinia genome, it is a harmless, but it will elicit the production of antibodies against the what? That is the what? Against the influenza virus, herpes virus, and hepatitis virus. So this is a broad spectrum vaccine which can be used against the what, that is a this vaccinia virus, hepatitis virus, against the what, that is a hepatitis B virus, herpes virus, and the what, influenza virus. So this is the what, the way by how by which the what you can produce the vaccine by the recombinant DNA technology, or the you can produce the vaccine. This is the same thing, that is a vaccinia virus surface glycoprotein, and you have isolated the gene for these three and now incorporated into the what, vaccinia genome, and again this is incorporated into the what in the vaccinia virus, and after that this will confer the what, that is this on the body. Here only the what, the surface protein is of glycoprotein, or that is the vaccinia, and but here you can observe on the surface, that is all the four what, that is the glycoproteins are there, and this is the what, the polyvalent vaccinia virus code, or this is a polyvalent, that is a vaccine by using the what, that is a vaccinia virus code, or you can prepare, or the you can confer the what, that is the immunity against the what, that is a many such diseases. So this is how you can produce the what, the immunity against the what, that is a that many viruses, or you can produce the vaccine by using the what, recombinant DNA technology. Thank you.